This is Talon, and today I will be teaching you about the cell cycle. So the cell cycle is composed of two phases, interphase and mitosis. In interphase, there are three subphases, G1, S, and G2. In the mitotic phase, there are four subphases, prophase, metaphase, anaphase, and telophase. The cell cycle is also run by triggers, such as the G1 checkpoint, the G2 checkpoint, and the M checkpoint. There are external and internal triggers. And we will also be discussing MPF and PDGF. In interphase, G1 equals growth. This is when the cell begins to grow and carry out its basic functions, such as synthesizing proteins or acting as a skin cell, secreting out sweat or other oils. During S phase, this is when the cell starts to prepare for mitosis. This is when it synthesizes replicated DNA. It's when it takes one strand of DNA, like so, and changes it into two. G2 equals preparation for mitosis. During this phase, your cell is growing to its final size, and it's carrying out everything it needs to do before mitosis begins. So prophase, the very beginning. This is when your cell this is when the nuclear envelope begins to dissolve, and when your chromatin begins to coil. Metaphase is where the normally recognized chromosomes are tightly coiled and aligned. They're aligned with the equator of the cell, ready for anaphase. Anaphase is where the chromosomes are separated, pull away from the duplicate. The chromosomes are headed for opposite ends of the cell. Telophase is where the nuclear envelope fragments that have previously been dissolved come together on both ends around the chromosomes. The cell is now being pulled in opposite directions, and two new, different nuclei are being formed. Now, here are your triggers. The G1 checkpoint is the first and most important checkpoint, as if it doesn't pass this checkpoint, it will never make it to mitosis. If the cell does not make it past the G1 checkpoint, it will enter a phase called G0, where the cell does not divide anymore, and it stays as it is. The G2 checkpoint is making sure that everything is ready for mitosis. It's making sure that the cell is at the right size and that it's healthy and it has all of the synthesized proteins secreted and ready for mitosis. The M checkpoint is a checkpoint during mitosis. Now, cyclin. Cyclin is a protein produced by the cell during a certain time of the cell clock cycle. Going back to here, cyclin is produced during interphase, and its level slowly and steadily rises until it gets to the mitosis, M checkpoint, where it drops off and it goes away. And this is a CDP, a cyclin-dependent protein. These proteins need cyclin to activate. If they don't have cyclin, they can't activate, and the cell will cease to carry out its function. More triggers. So this is mitosis. So what I told you, the MPF and cyclin dependency is an example of internal triggers. So during mitosis, you see that MPF and cyclin have reached its highest point before it sharply drops off at the start of interphase, where it slowly begins to rise again. And once it gets to mitosis, then it drops. Rises once again during interphase and mitosis. Then once it reaches the peak of mitosis, it drops. An external trigger would be anchorage dependence or density dependent inhibition. Density dependent inhibition is when cells grow along a single plane and they don't grow on top of each other. And once they get to a certain density, when everything is touching each other, they cease growing. But if you were to take out a few cells, they would regrow, as seen right here. They regrow until they're touching again and do not overlap. This is what normal healthy mammalian cells look like, showing density inhibition. But these are cancer cells, as they show no sense of density inhibition. Cancer cells are formed by cells who ignore the triggers. It's internal and or external. For example, these cells are not showing density inhibition. They continuously grow upon one another, growing in size and starting to invade other tissue. As you can see, they are going to start invading the bloodstream. This means that they will continue multiplying with a non-determined stopping point. If they do stop growing at all, this creates a tumor. This is what normal mammalian cells look like. They start to grow and they show density dependence, and they're not all growing on top of each other with no abandon or sense of direction. And that's the end of the slideshow. I hope you learned something, and please give me an A.